Good morning or good afternoon, everyone. There's a few people still arriving, so we'll just wait a few seconds for everyone to arrive and then we'll get started. Okay, I think we'll get started. Welcome everyone to the CanPath research webinar. My name's Trevor Dummer. I'm the National Scientific Co-Director of CanPath, the Canadian Partnership for Tomorrow's Health. And welcome, thank you for joining our webinar. Before we get going and I introduce you to our speaker for today, I'd like to just start with a land acknowledgement. We acknowledge the traditional territories of the Mississauga of the New Credit First Nation, the Anishinaabe, Wendat, Huron, and Haudenosaunee Indigenous peoples on which CanPath National Coordinating Centre at the University of Toronto resides. The territory was the subject of the Dish with One Spoon, Wampum Belt Covenant, an agreement between the Iroquois Confederacy and the Confederacy of the Ojibwe and allied nations to peaceably share and care for the resources around the Great Lake. CanPath teams work across Canada from coast to coast, and we acknowledge the ancestral territories that are home to many Indigenous peoples from across Turtle Island, and we are grateful for the opportunity to work on this land. Personally, I'm calling in from the traditional, ancestral and unceded territory of the Musqueam people at UBC Point Grey campus. The Musqueam people have been stewards of this land since time immemorial, and I gratefully acknowledge being an uninvited guest on this land. Given everyone's joining from across Canada and many different territories, there's obviously an opportunity for yourself to reflect on the land that you're currently residing on. So, to the webinar, welcome. As I said, we're shortly going to be hear from Dr. Sasha Bernatsky, a rheumatologist, James McGill Professor of Medicine at the McGill University, and a senior scientist at the Centre for Health Outcomes Research at the Research Institute of McGill University Health Centre. Dr. Bernatsky has many affiliations, including with the McGill Environmental Epidemiology Research Group, with CANU, the Systematic Lupus International Collaborating Clinics, and the Canadian Network for Improved Outcomes in SLE. Dr. Bernatsky has been a long-time collaborator with the CanPath team and has been an extensive user of the CanPath resource. So we're extremely excited to listen to Dr. Bernatsky's reflections on some of her research, particularly in relation to environmental exposures and rheumatic disease. Before Dr. Bernatsky's presentation, just a little bit of housekeeping for everybody. There's a Q&A button at the bottom of your Zoom screen. Please use that to enter any questions that you have for Dr. Bernatsky. We should have plenty of time at the end, around 10 to 15 minutes. And so we either will be able to answer those as she's speaking, or Dr. Bernatsky will be able to take those at the end. So over to you, Dr. Bernatsky. Thank you.
Thank you so much. So as I begin, I want to acknowledge two individuals, Naizu Zhao and Audrey Smargiassi, who are the methodologists behind some of the results I'll be presenting to you today. I'm a rheumatologist and an epidemiologist. So to start us, I know it's a varied obvious, so to make sure we're all on the same page, I want to talk for a minute about autoimmunity that underlines the autoimmune rheumatic diseases that I'm going to be talking about today. So we know that antibodies or proteins, our immune system, specifically the white cells, certain white cells produce to fight infection. So if our body detects an infection by a virus or a bacteria, our body makes antibodies against the pathogen. And autoantibodies are proteins that are mistakenly produced that target a person's normal tissue instead of protecting against infection. So that can cause inflammation and often damage. And that can affect the joints or the skin or the kidneys, a whole variety of different body parts. So I'll be talking today about rheumatoid arthritis, or RA, and systemic autoimmune rheumatic diseases, or SARDs. And these are characterized by these autoimmune diseases are characterized by specific antibodies. So in RA, it's the anti-citrulline protein antibodies, or ACPA, that are specific to RA. And in SARDs, they are characterized by the presence of anti-nuclear antibodies, or ANA. In the reality, there's a host of different autoantibodies as well, but these are the two that we're going to be looking at today. So why would somebody begin life healthy and then go on to develop RA? So you can see a photo there of someone who has uncontrolled rheumatoid arthritis that has affected the joints and caused disability. We want to understand how a person gets from before disease to actually clinical disease, because that would help us potentially not only help treat better, but maybe even someday come up with an effective prevention strategy. So everybody has some kind of genetic risk factors for something. And in RA, there's a multitude of different potential risk factors genetically. These aren't like a monogenetic risk factor. And even if you have some of these genetic risk factors, you don't necessarily go on to develop RA. It seems that there's stuff in the environment that's happening, such as smoking and other exposures, that can ultimately cause initially the expression of autoantibodies like ACPA in the case of rheumatoid arthritis, and eventually the clinical manifestations. So you can find these autoantibodies ACPA in the case of RA years before you actually develop clinical manifestations of RA. And in the SARS, it's believed to be the same sort of scenario. So if you take as an example systemic lupus or SLE, which is one of the prototypical SARS diseases, this is a disease that can affect many different parts of the body, skin, joints, and even kidneys. Sometimes these people require dialysis because of kidney damage. So here we have, again, the kind of multitude of potential genetic factors not completely understood or worked out, and with also the requirement for there to be some kind of environmental insults to end up with clinical manifestations of systemic lupus. So and other related diseases. So for SARS, ANA is the type of antibody we'll be talking about today. So one of the reasons I embarked on this area of research is that we knew that there were different environmental exposures, but things like occupational exposures really didn't explain the majority of RA or systemic lupus risk. So there's, traditionally things like respirable silica or occupational exposures to solvent we're seen as risk factors, but the majority of people don't have those risk factors. The majority of people who have lupus or RA don't have those risk factors. So this made me think we have to look to another set of exposures. And I thought of air pollution, because we're all exposed to air pollution to different extents. And air pollution is very complicated. So we have a combination of different gases. I've listed some here, like sulfur dioxide and nitrogen dioxide, and also particles. So these are actually quite complex mixtures of particles and droplets, and they have varied characteristics with respect to surface area and chemical components and their solubility. But one of the most important particulate matter health exposures is PM 2.5. So this is very small particulate matter, less than 2.5 microns in diameter. And we can see the picture here shows a human hair, and something like a speck of pollen is very much smaller, which is represented by the blue spheres. But PM 2.5 is even smaller, and that's represented by the pink spheres. PM 2.5 itself is made up of chemical components to varying degrees. So you'll have makeup of PM 2.5 represented by sulfate, nitrate, ammonium, black carbon, organic matter, mineral dust, and salt. And from one area to another, the chemical component the amount of each of these can vary quite a bit, and that can affect potentially their effects in terms of health. But in general, PM 2.5 is important because it's small enough that when you breathe it in, it can get all the way to the end of your airways and cross over into the bloodstream and cause inflammation. And although we do have pretty good air quality in Canada and in North America, it's important to remember that over 85 million North Americans still live in areas where air pollution exceeds air quality. So this is a very crude cartoon where I've pulled together, some of that thoughts on why PM 2.5, when it enters your bloodstream, could cause different parts of your immune system to be overactive and maybe trigger the production of autoantibodies and perhaps actual disease. So to try to address this, we turn to CanPath. And many of you know CanPath as the general population cohort of over 300,000 Canadians. And they're each, the provinces in Canada contribute to it with their own cohorts. So what we started with was looking at the baseline sera for regional CanPath general population cohorts from Quebec, that's Cartagena, and the Ontario Health Study. So not everybody who's a part of these cohorts donates a blood sample at the cohort entry, but those who do, we were able to use. So we also have, of course, information that individuals provided cohort entry on demographics, medical history, and residence. We randomly chose over 7,000 subjects with available baseline sera from Cartagena, and we have also analyzed over 3,000 from the Ontario Health Study, and we've assessed sera for ANA and ACPA. So we have, to assign the air pollution exposures, we work closely with CanPath and provincial data holders. So some of the results I'll show you today will also include provincial health data analyses. So working with the data holders, we assigned every subject values for their different environmental exposures that we'll be looking at based on the 6-digit postal code information. So I'll mention that we are also next looking at sera from BC Generations Project and the Alberta Tomorrow Project, but I don't have results from that to show to you today. So how do we get the information on air pollution exposures that people are subject to? So here is a map of the National Air Pollution Surveillance System with the red dots are where ground level monitoring stations are. So these can sample the air and tell how much PM 2.5 and other air pollution components are in where the monitoring station is. But you can see there's lots of areas where we don't have data on air quality. So what do we do? More and more, so I think the standard now is to use satellite imagery estimates for air pollution exposures. So we have the monitoring stations, but we methodologists have taken satellite data. So you have images and imagery information that can be correlated with the measurements at ground level. And so the methodologists have modeled what data collected by the satellite represents what extent of air pollution at the ground level, and then used other information on topography or weather or so on to estimate throughout time and over a much larger area of Canada or other countries that have these estimates available. So this is some of the people behind this, behind making these kind of estimates available to researchers is the Canadian Urban Environmental Health Research Consortium, or CANU, and I think we have some people from CANU on the call today, so that's much appreciated. So the first thing I'm going to talk about is an analysis we did using the Ontario Health Study cohort and data. So we looked at serum samples from over 3,500 subjects, which we randomly selected from amongst all the people who contributed a sample. And then we look at ANA titers. So I'm going to say this is how I practice explaining it, but I see Marvin Fritzler's on the call as well as other rheumatologists, so they'll probably laugh at how I'm going to be explaining this, but this is how I was trying to convey the idea of a titer of ANA antibodies. So you use immunofluorescence assays to look at sera that's been diluted, and then you continue to dilute it to see how strong that signal is. So if you detect antibodies by immunofluorescence at when you diluted that sample to 1 in 80, that's not very strong. But if you're able to see that immunofluorescence as you go further in dilution up to 1 in 320 or higher, then it becomes evidence of a more robust autoantibody response. So as I mentioned, we're looking at ambient PM2.5 estimates, and those were obtained by the modeling that I just described, which other people have made available. So then this is a cross-sectional analysis because we have a single sample per person that we're looking at. And we computed the adjusted odds ratios for ANA positivity. And for our exposure of ambient PM2.5, we looked at the highest quartile of exposure to the lowest quartile of exposure. And we adjusted for age, sex, race, ethnicity, smoking status, and the rurality index of Ontario, which characterizes urban versus rural status, because as we said, the components in PM2.5 can vary quite a bit depending on where you live. So when we compared the highest versus lowest quartile PM2.5 exposures, the adjusted odds ratios for ANA titers of 1 in 640 was 1.46. And for titers of 1 in 1280, the odds ratio was 1.54. Lower titers weren't clearly associated with PM2.5 exposures. So we said overall, it seems like there's some evidence that PM2.5 exposure was associated with ANA positivity, at least at high titers. And this could strengthen the argument that air pollution could have systemic immune system effects that could in turn lead to autoimmune rheumatic disease. So next, I'm going to tell you about something we published a couple of years ago on systemic autoimmune rheumatic diseases and industrial air pollution emissions. So this was using administrative health data from Ontario. We had assembled an open cohort of over 12 million adults without SARS diagnoses at cohort entry and followed them until SARS onset, which was our outcome of interest, or death or emigration or end of study. So we used physician billing codes and hospitalization diagnostic codes to identify SARS. And in Ontario, the physician billing codes are such that you can't identify systemic lupus or scleroderma or myositis or the others specifically. They're all under the same billing code. So we looked at average PM2.5 nitrogen dioxide and sulfur dioxide industrial emission exposures every year up to 1 year before either SARS onset or end of study. And we assigned these using residential postal codes. So as I mentioned, we can't have access to postal codes within the administrative health data because of confidentiality issues. So they worked with us to assign the exposures, and then they gave us the data set without the postal codes, and we analyzed it. So here we used a different approach where we're considering 3 different air pollution exposures and they're correlated. So we want to model them. Here we're modeling time to SARS onset, but a simple survival analysis like a Cox proportional hazards model, it would be difficult to put all 3 exposures together because they are correlated. And when you put correlated variables in a model like that, you can end up with misleading or results that are difficult to interpret. So we used a quantile decomputation model for time to SARS onset, and that looks at the 3 exposures of interest increasing in the quantiles at the same time. And we adjusted for sex, age, regional income, the rurality index, and calendar year. And we didn't have information on smoking, but we adjusted for COPD because other analyses of environmental factors using administrative health data have done that as a proxy for smoking, given that COPD and smoking are so closely correlated. And we also looked at ambient PM2.5. So we found almost 44,000 new SARS diagnoses, and the adjusted hazard ratio for SARS onset related to an increase in those 3 industrial emissions by 1 decile was 1.018. and so in these type of analyses, you can, you also are provided with information on which of your exposures contributed more to risk. So our three air pollution exposures, PM 2.5 seem to contribute more to SARS onset than nitrogen dioxide or sulfur dioxide in terms of industrial exposures. So here we have also found positive association between SARS onset and some elements of air pollution. So what's the story with ACPA and RA? So we have looked at ACPA in almost 8,000 randomly selected general population subjects from Cartagena. And ACPA levels can be described in terms of different thresholds. So a threshold of 20 units per mil would be more of a low positive, with a 40 being maybe a more moderate positivity and 60 being a higher positivity. Here, we were looking at industrial sulfur dioxide, nitrogen dioxide, and PM 2.5 concentrations estimated by the California Puffin atmospheric dispersion model. So that's another form of modeling. So again, methodologists have made estimates for all different postal code areas using information about where these industrial emissions are coming from and atmospheric conditions and other variables to be able to estimate the exposures for all postal codes throughout the region you're interested in studying. So here we looked at some single exposure logistic regressions for ACPA positivity. So looking, for example, at PM 2.5, industrial PM 2.5 on its own. But that's, again, we said it could be a bit misleading because there's other air pollution components that you should be counting for at the same time. But if you try to put all three in a simple model, it could cause problems. So we did a similar weighted quantile sum regression. So we had the three industrial exposures, the outcome of ACPA positivity, and we're increasing those three air pollution exposures at the same time when looking for effects. So here we adjusted for age, sex, smoking, and family income. And we also looked at regional ambient PM 2.5 exposure. So we found with this, in this simple single pollution logistic regression models, we found with ACPA positivity at 20 units per mil, the odds ratio for PM 2.5 industrial emissions was 1.19, and for sulfur dioxide, the odds ratio was 1.03. We didn't see clear associations for nitrogen dioxide. So these were the single pollutant models. In the weighted quantile sum models, where all three were present at the same time, there was a positive association between an increase in all three of them together and ACPA positivity at the 20 unit per mil threshold. And again, in that model, there's information about the probable weight of each of your, in this case, three exposures that you're looking at jointly. And so that information suggested that industrial PM 2.5 may have the closest association with ACPA positivity as opposed to sulfur dioxide or nitrogen dioxide. And so we didn't see as strong associations between ACPA and regional ambient PM 2.5, interestingly. So here we also seem to have some suggestion of positive associations between ACPA and industrial emissions of PM 2.5 and maybe sulfur dioxide. So what about RA onset? So here are analyses where we looked at Ontario Health data again, an open cohort of adults without RA, following them up until RA onset, which was now the outcome of interest, or death or immigration from Ontario, or end of study. And incident RA cases were identified again by physician billing and hospitalization discharge diagnostic codes. Here we looked at average levels of PM 2.5, so ambient levels of PM 2.5. And we did look at overall ambient PM 2.5, but then we also broke it up into the components. So as was mentioned, all PM 2.5 isn't necessarily equal. It may be that the PM 2.5 from Sudbury, Ontario is very different from the PM 2.5 in Regina, Saskatchewan. So you can get information. It's available again from the methodologists who produce who make these exposures accessible. You can get information on the breakdown of the PM 2.5 in different postal code regions, and we assigned those exposures to individuals for the 5 years before cohort entry. Again, so we did use the simple Cox hazard model for time to RA onset, looking at just a single exposure of ambient PM 2.5 as itself, but then we also looked at a quantile G-computation model where we could include those different components of PM 2.5 and increase them all together as we look at the outcome of RA onset. So we identified almost 68,000 new RA cases, and the adjusted hazard ratio for time to RA onset from the for overall PM 2.5 was 1.023, and it was 1.027 for every decile increase in exposure to all of the different PM 2.5 components. And again, this model can give you information about which of the components seems to be contributing the most. And among the components, ammonium seemed to be contributing most to RA onset. So if I have the moderator will have to tell me if I'm getting close to time, but I thought I could spend a bit of time next talking about our next project. So last year we were funded to look at PM 2.5 related specifically to forest fires. So the idea is that climate change is one of the factors in increasing Canada's wildfire season. So 2023 was one of the worst, well, it was the worst wildfire year since 1995, with just millions of acres of forests being burnt across Canada. 2024 was almost as bad. It ranks as the second worst forest fire season since 1995. And wildfire smoke, of course, contains large amounts of air pollutants, and many of us kind of experienced especially in the summer of 2023, what that looked like in terms of the quality of our air. So as other sources of air pollution are increasingly regulated, wildfire smoke has become an even important source of ambient 2.5. So if industrial emissions and road traffic sources of PM 2.5 are being more regulated, wildfire smoke PM 2.5 might be increasingly important. So the other factor going on is that climate change leads to more extreme temperatures and temperature variability, and those potentially could have some adverse effects on our health. So you have to think about both of them as you're trying to determine, can we study the effects of PM 2.5 from wildfires on autoimmune effects such as RA or SARS or their autoantibodies. So this, again, the new project will have some objectives looking at the autoantibodies and using CanPath sera, and then the other component will be, again, using administrative health data to look at the incidence of RA and SARS. So these analyses will look at wildfire PM 2.5 concentrations, and will also adjust that for ambient temperature extremes and variability and other factors. So to get to the PM 2.5 exposures related to forest fires or wood or biomass burning, we have the Canadian Optimized Statistical Smoke Model, which was developed by the Environmental Health Services of the BC CDC, which estimates PM 2.5 exposures more specifically for wildfire smoke. So again, this uses some information about satellite imagery with some ground monitoring information, and breaks up the results into the warmer seasons and the colder seasons. So in the warmer seasons, that's when these estimates reflect wildfires, and in the winter seasons, it's actually wood stoves that are contributing. So we will look at the warm seasons to try to get the picture of what PM 2.5 estimates for wildfires, what effects they have on either antibody, presence of autoantibodies or RA or SARS incidents. So this just shows you that the satellites can tell where the wildfires are because of smoke plumes and also radiation of the fire. And they combine that information with the aerosol optical depth imagery, which is the amount of light that's being absorbed by particles in the air between the satellite and the ground monitoring stations. And they do the same kind of analyses to estimate PM 2.5 exposures from smoke for all regions, all postal code regions. So then temperature extremes and variability. Now the body's pretty good at maintaining core temperatures, but even when there are changes in ambient temperature, but the core temperatures can still be affected to some degree, especially for older individuals who may be more at risk for RA and some SARS. We'll get the mean air temperatures from atmospheric temperature estimates, and we'll be looking at variability as well, because temperature variability as well as temperature extremes could be important for health outcomes like the ones we'll be considering. And then we have to think about a relative humidity, because humidity does have an effect on how people experience rheumatic disease or joint symptoms. So we'll look at that. And then there's other things to think about, like UV light, which can be a trigger for some SARS, or viral infections, which can be triggers for SARS. And we do have self-reported sun exposure for CanPath subjects. For viral infections, we'll be limiting our primary analyses to warm seasons. So that's when most endemic respiratory viruses are in low circulation. But we'll also think about, calendar month variables to when we're doing analyses, sensitivity analyses, evaluating year-round outcomes. And we're not using, for the serology anyway, we're not using data after 2019, so COVID-19 won't be an issue. So same thing as I mentioned for using administrative health data for to look at RA and SARS onset. So that's really what I have to present today, that RA and SARS are important autoimmune diseases, and their unclear etiology remains a key knowledge gap. CanPath sera and sample and data were extremely helpful. In fact, I just don't see any way we could have done what we did without CanPath. And our analysis with population-wide administrative health data helped us bring the two sides of the coin together. So on one hand, the autoantibodies, on the other hand, the incidence of RA and SARS. So there are a lot of people who made this happen. So I hope I didn't forget many people here, but I'm just acknowledging some of the major players who helped us get this started and helped us see us through to the results that I presenting today. As mentioned, we'll be also working with the BC Generations Project and Alberta's Tomorrow Project. And again, Audrey and Dr. Vina Zubieta from UBC, Dr. Chen from Health Ontario, May Choi and Marvin Fritzler, who performed the autoantibody analyses for us, and Autumn Neville and Jennifer Lee, who just made things happen. So thank you so much.
Thank you, Sasha. That was excellent. It's nice to see some of those results, because I can think back to the many years ago. CanPath has been going for 15 years or whatever now. It's a considerable amount of time, and I recall the early collaboration. So it's great to see the fruits of all of those labors. So we do have invite everyone with questions. Please do use the question and answers, the Q&A in there to type in. We've got plenty of time for questions. There's a couple already, so just going to go through them from top to bottom. So first question is from an anonymous attendee. Is there biological evidence or a proposed mechanism explaining how PM 2.5 exposure leads to increased autoantibody levels?
Right. So there is some basic science data to suggest the different mechanisms. So some of them I kind of put in this horrible cartoon. I'm not a basic scientist, so people who know more than me, this is hand waving in one slide. So, but there's the in different disease states, people have looked at different parts of the immune system, and these all work together to, in general, stimulate our white cells and could potentially lead to auto antibodies. So what can you measure? So PM 2.5, definitely, if you look in the lab, you can expose there are lab experiments that can show how PM 2.5 can stimulate the generation of reactive oxygen species and upregulate genetic factors that keep the immune system activity going. PM 2.5, I think there's growing evidence that PM 2.5 definitely has a systemic effect on the body. I'll say one of the really cool things about these kind of analyses, it was the first, prior to these kind of analyses, we had, good data on what happens when you are exposed to PM 2.5 in terms of asthma risk or asthma exacerbations or other cardiorespiratory disease or lung cancer. And that makes sense because you breathe in the air and there's like a local effect, one would think. But this was one of the, I think these kind of analyses where you're looking at a totally different kind of disease, it's convinced, I think, some of the people who made policies for Canada on air pollution levels, that indeed you can have health effects far beyond just the lung. So I guess that's all I have to say.
That's great, thanks. Next question from Janet. Has there been monitoring of rural farm areas for pollution from agricultural chemicals, which are often applied by crop dusters?
Yeah, so I think a lot about that because I grew up in rural Saskatchewan. We always saw the airplanes overhead, the farm planes to distribute pesticides or whatever. And so as we've discussed, the monitoring stations are not everywhere, right? So if you look to where the monitoring stations are, they're not necessarily going to cover rural areas. I mean, you can see how few there are for Manitoba or Saskatchewan, but you can use these wonderful methods. I'm really grateful to the methodologists who are able to estimate what exposures would be in the rural areas. Now, there's a little bit more difficulty, I would say, in the rural areas because you have fewer postal codes. So people who are, averages over postal code regions, but in the rural areas, those postal code regions are going to be really big. So that's a challenge. When it comes to, what are the, effects of agricultural activities on the quality of our air? It's interesting because we mentioned in that one analysis where we looked at PM 2.5 components, that ammonium was one of the components that seemed to be most important for perhaps being linked to autoimmune rheumatic disease. And that has also been shown for other inflammatory or autoimmune conditions like interstitial lung disease, and even in interstitial lung disease that develops in people with RA. So ammonium does come from agricultural practices. So I feel like as we drill down deeper into some of our analyses, we can learn about the effects of agricultural practices on our health, but there are definitely challenges to get there. I hope that answered a little bit of your question, Janet.
Great. A question here actually from Charmaine. Does this association explain the greater incidence of RA and lupus in First Nations people seen in Canada and Australia? There's much higher incidence in these populations. And have you been able to look at that?
Yeah, that's a great question. So, you know, what would be great to do, and I would say probably the people in Canada that are best positioned to understand better about First Nations health risks, including RA and SARS, would be there are researchers doing wonderful work in Manitoba, where we know there's a lot of First Nations populations that are affected by diseases like RA, and they have for years been developing good relationships with those populations to do meaningful research on what's driving the risk of those conditions, but to do it in a way where you're really engaged with people, and so that's one of my hopes, is that those kind of relationships will help us understand whether air pollution, especially air pollution generated from things like wildfires, are impacting First Nations individuals. So I think also BC has the potential, for that also. We shouldn't be doing that for every province and jurisdiction in Canada, but it's not an overnight process, because there have to be those relationships with the people in those communities. So that's how I see it. But I do hope that this kind of research helps move those challenges along.
Yeah, absolutely. And I think part of it as well is, as you've already mentioned about they're monitoring stations, but they're not everywhere. They miss out rural areas. Then, we need also much better monitoring stations so we can model better. So we can look at, more specific geographic areas. This actually links to the next question from Marvin. Are you able to look at PM2 in specific geographic areas, for example, the oil sands? So it's talking really about how locally can you go?
Yeah. So one of the tough things, I know that people have tried to try to identify hotspots. So Janet Pope, a number of years ago, I believe she published on mapping cases, and I think it was cases of systemic lupus, but I'll have to look it up. But she did some modeling there to try to find the hotspots. So the problem is when you have disease incidents like systemic lupus, well, the prevalence is 1 in 2,000. The incidence is much smaller. So it's hard to have robust, to produce, research that has robust estimates of a disease incidence that is 1 in 10,000 or something like that, when it's a small area. So that's one thing to consider, but that's definitely, one of the things we started out doing years ago was trying to think of, it's not just air pollution, right? It's also like water quality, so, and many other things. So that's a whole bunch of other challenges that it arises because of the relatively low incidence of these conditions.
Great, thank you. Okay, turning now, I think a slightly more technical question from Christopher. Can you discuss the rationale for operationalizing PM 2.5 as a discrete variable with cut points at empirical quartiles? What are the advantages to this compared to other methods for modeling continuous air pollution outcomes, such as splines, GAMs, etc.?
Yeah, that's good. So I think modeling splines can be helpful. We have done that. We've looked at PM 2.5 in different ways. So we have looked at it continuously, but that's probably not what you want to do, because there's probably thresholds. Not everything is going to be kind of linearly related. So we've been doing this for a long time. Like the earliest, our earliest analyses like 10 years ago were probably much cruder, but we were learning, right? So we don't know what's the best way to model these things. If you look if you look, I think, as I mentioned, spines is nice because you're trying to reflect what the data are telling you. I don't know if we need to worry a little bit about overfitting when we do that, but I mean, there's methodologic ways to try to limit that potential downfall. So I think, so some of our analyses now are using splines. But I feel like we don't actually know the best way to model these associations. But I hope that we're learning. But I do think probably there's some kind of threshold effects.
Okay, thanks. Do other systemic autoimmune diseases beyond rheumatic conditions also show an increased risk associated with higher PM2.5?
So so something like inflammatory bowel disease would be a good disease to study. I'm not sure of the most recent evidence. I'm pretty sure somebody has looked, but that would be another condition that could be helpful to study. Some of the more rare autoimmune diseases like myasthenia gravis, like they're even a magnitude, I think, less common. So that would be tough. I've seen researchers using administrative health data, trying to look at a whole bunch of theoretically autoimmune conditions together, including diabetes, like type 1 diabetes, and other things where, yeah, it's an autoimmune, like autoimmune thyroid disease. Like, yes, there's an autoimmune component, but I guess the diseases, as a rheumatologist, you're used to seeing, like lupus and rheumatoid arthritis, they're so linked to the autoantibodies that I think it's a really good test case. So there's no reason why not to think that the other conditions could also be associated with air pollution. But for me, these rheumatic conditions are cases where we have autoantibodies that are pretty good markers for disease state. So you're able to, like I said, combine the antibody analysis with the incidence analyses. And I don't think that you really have that for something like inflammatory bowel disease and some of the other things. So it'd be good to study, but I think in rheumatic disease, it's a good disease domain to look at this question.
Thank you. Okay, moving, it's a bit more of a knowledge translation type question. Regarding risk of exposure to PM2.5, say to RA as an example, interpretation of risk can be challenging for lay people and policymakers. Have you worked to try and express the increased risk in other ways that may be more accessible? I guess this moves towards the sort of prevention side as well, in terms of advice.
Right. So, in general, there are tools that anybody can access. Like, so if you have a computer, if you have internet access, you can find out about the air quality index of the day in the region where you live in Canada. And other countries have similar air quality indices. And in fact, there's an international group that would provide air quality index values for wherever you're living. For most populated places. So you can know what the air quality is. Those websites tend not to have any kind of specific recommendations for like what risk does it present to somebody who's either at risk to develop RA or who has RA and is worried about a flare. We don't really have that kind of detailed information, I think, on how likely is a certain level of PM2.5 exposure going to increase your risk of a lupus flare. There are a couple of studies in systemic lupus that do show associations between air pollution like PM2.5 and certain components of lupus activity. So to me, that's again a good indicator that this is a real phenomenon on the immune system, but it doesn't allow us to necessarily say if the air quality index is such and so that you have, a twofold or fivefold increased risk of flaring if you go out. The other thing is, what can you really do to protect yourself? There are some things you could, right? So say you have interstitial lung disease, which is, you've got structural issues too, because this autoimmunity has affected your lungs. So you go and you look at the air quality index. Well, there are some things you can do to protect yourself. If it's the summertime and you were thinking of being outside, you could stay inside if you have air conditioning. But if you don't have air conditioning, you have to have your windows open, then, you know, there's a bit, I feel like maybe there's a bit of limitation of how helpful the information can be to people who need the information. But, you know, I guess what we need we currently aren't developing tools for RA patients. I will point to, because I didn't yet point out that an international group of rheumatologists, here we go. It's called React Rheum. So there is information on this website for people who are interested in rheumatic diseases and the effects of the environment, including air pollution. And so there are some tools and links to website that try to provide answers for people. But there's still work to be done. There's it's part of our wildfire smoke grant from CIHR. We are working with a group of really good knowledge translators, including Danny Blair in Winnipeg, who has a beautiful website about environmental, climate change, environmental exposures, air pollution, and health. So as we did have knowledge translation funds to try to put information on the website, which has beautiful videos and interviews with people, to make that knowledge at least accessible, even if we don't have perfect solutions for people.
That's great. I wonder if at some point, you know, if you've got a phone with an app and it's got a monitor and a sensor on there, if you could actually interface some of that online information with some, you know, real time, you know, maybe the data's not there yet, but in the future that could that give some advice to patients about their exposure and what they might actually want to try and do to mitigate their, you know, immediate risk of a flare.
Right. So there was a beautiful project I attended at. It was a student who did a project in a rural community that was subject to forest fires and a lot of First Nations people. And they worked on messaging for that community and made it very specific to their needs, like using the local radio station and other forms of, you know, messaging. So that I really like that as an example of knowledge translation and trying to give people the information they needed.
That's great. I think we've got time for a couple more questions before we have to finish off. So I'm not sure we'll get through them all, but we'll do our best. From Hamad, is there a link to environmental pollutants causing disease flares in these patients, or more so with triggering the disease process in conjunction with genetic risk? A bit of a sort of a, you know, is it genes or environment or an interaction?
So I mentioned in lupus, there are some clinical studies where people have information. In fact, way back, we did this for our lupus cohort in Montreal, and we did show variation in PM 2.5 was associated with some disease outcomes like renal indicators and serologic indicators of lupus activity. And the Johns Hopkins lupus cohort also did a good job more recently of showing those kind of associations. We've looked at other disease. We looked at scleroderma and air pollution years ago, and we didn't see signals there. But other people have used administrative health data analyses to look at PM 2.5 and things like admissions for lupus and found associations. And I think in myositis, there's at least one study that showed that as well. And there's a related question there, I think. Oh, genetic. Okay, so there was
genetic.
Was a paper published recently, past year, I think, about genetics and like a polygenetic risk for lupus and air pollution, which supported the concept that people who there may be some genetic risk factors that places you more at risk when it's subjected to air pollution to develop disease. And that matches perfectly with our way of thinking of these diseases, that not everybody will get these diseases, but the combination of genetic factors and environmental factors leads to disease. The problem is that we don't know everybody's genetics right now, and the complex of these diseases, the genetics behind these diseases is very complex.
Okay, another question from Alan here. Do you have any thoughts about the role that prenatal maternal environmental exposure might have on influencing future
That question comes
autoimmune?
from Dr. Rosenberg. So hello. I was happy to see your name there. So wow, what a good question. So Dr. Rosenberg created the Bebop cohort, which was a great, like, collected amazing information to try to get to the answer of what puts people at risk for autoimmune rheumatic diseases, especially the ones that emerge in childhood, because there's a pretty significant genetic loading, because earlier you get a disease, probably the higher your genetic risk factors are playing in there. So there have definitely been studies of air pollution and JIA or pediatric onset lupus. So is it more important or less important in kids versus adults? Well, our hypotheses that in the peds onset cases, it's more of a genetic component, but I think both are probably, I think we embrace the concept that you need genetics and environmental exposures.
That's great. So thank you, Sasha. I'd like to thank you for the excellent presentation and answering all of those really interesting questions. And thanks everyone for joining us and all your questions, your interest. That's about all we've got time for. So I apologize if we didn't get through to your question. Please reach out through to CanPath. I'm sure Sasha will be delighted to try and answer some of those questions online as well. But please do connect. So just wanting to say, if you're interested in finding out more about any environmental exposures, I believe there's some information in the chat, links to the website, the canoe data, the environmental data that's linked to CanPath. If you're interested, please do join us for our next webinar. I believe it's on March 11th at 11 a.m. EST on genomics. So looking forward to seeing you next time. Thank you very much. Thank you, Sasha, once again.